So now I'm going to discuss with you about the initiation, elongation, and termination process of translation both in prokaryotes and eukaryotes. And let us get started now. So normal, the figure that you can see here, that you are seeing here, is actually the ribosomes and the overview process of translation process, how what happens when translation process occurs. So at the first you can see ribosomes are getting assembled. The ribosomes are getting assembled. The small ribosomal subunit are associating with the mRNA, and they are associating with the mRNA within the ribosomal binding site, and along with that the initiator tRNA also comes, but it does not occur in that way. Some other associated translation factors come into play for this initiation process, which allows the large ribosomal subunit to bind. And after that, after full assembly of the large and small ribosomal subunit, the initiation factors get shed off. It removes out. In that place, the elongation factors now will be used for the elongation of translation process. Now you can see that in the next figure, the ribosome is moving from 5 prime to 3 prime direction in order to synthesize polypeptide. Well, the polypeptide which is going to be synthesized, it's the N terminal end, and at the C terminal end they are incorporating new amino acids, and this polypeptide synthesis is catalyzed by the 23S rRNA of the large ribosomal subunit in case of prokaryotes. Okay, and each time this ribosome is identifying a codon sequence which allows incorporation of a newly charged tRNA known as amino acyl tRNA carrying a specific amino acid for that particular codon, and catalyze this peptidyl transferase reaction that I have shown it to you. At the end of the transcription process, at the end of the translation process, what happened during the end process, it recognizes the stop codons are being recognized, and after recognition of the stop codon, the ribosomes then again disassemble, and you can see the polypeptide chain is also got off from the ribosome, and all the other, and then comes a termination factor. Now for termination of the translation process, termination factors then play an important role for the translation process. Okay, and you can see that they getting disassembled. So I was talking about the termination process, overview of the whole translation process. That during translation, termination, the termination factors then comes after after the ribosome identify the stop codon. Here you can see the stop codon, which is shown here, and it recruits the termination factors like ribosome recycling factor. Okay, it also recruits the release factor 1 and release factor 2 to allow the termination of the translation, and due to which what happened, the ribosomes then disassociate from each other. Disassembly occurs so that they can carry out the next round of mRNA. They carried out the next round of translation process, and the polypeptide chain is now destined for further modifications known as post-translational modifications. So in this way, you can see that how the ribosomal subunits are assembled at a certain position on the mRNA. Well, we all know that first, a small ribosomal subunit bind to the mRNA using the 16S rRNA, which complementary binds with the ribosomal binding site, also called signed Dahlgren sequence, found within the prokaryotic mRNA on the upstream. In case of eukaryotes normally eukaryotic ribosome binds by identifying the 5 prime cap. So by observing the 5 prime cap, eukaryotic ribosomes identify that 5 prime cap and bind. Okay, then you can see that after assembly, now this is not a simple process because it needs some other transcription translation factors to come and bind. Okay, both in case of prokaryotes as well in case of eukaryotes. In the elongation process, you can see the ribosomes are moving from 5 prime to 3 prime direction and is translating the polypeptide chain. And during translation elongation process, ribosome actually identify one codon at a time. So it identifies, recognize one codon at a time, and then it recruits that specific amino acid tRNA for that particular codon that carries that amino acid and carries out the peptidyl transferase reaction. Now, peptidyl transferase reaction is catalyzed by the 23S rRNA by the large ribosomal subunit in case of prokaryotic translation process. Now, in this way, the translation goes on, and here also elongation factors play an important role. Elongation factors, there are two types of elongation factors you will see later on when I will discuss them elaborately. And then in the at the end, you can see that when the ribosomes encounter a stop codon, then it recruits the termination factors like ribosome recycling factor, like your release factor 1, release factor 2. Okay, so to terminate the process, and then the ribosomes are disassembled again. Polypeptide chain are also getting disassociated from the ribosomes and are now destined for the post translational modification. Okay, and the ribosomes are now destined for next round of translation synthesis. So in this way, your whole translation process works. In an overview, if I say, this is a polysome that has been shared before, and theoretically you can see in one mRNA multiple ribosomes have been associated with it and carry out the polypeptide chain synthesis. This normally observed on the rough endoplasmic reticulum. If your protein, the peptide, is your related with secretory function, having some secretory function, then only this type of things, this type of polysomes can be observed. Now, one more thing that I want to wish to say when I begin that process of translation is that ribosome have three sites. Okay, ribosome have three sites which where it recruits the factors, it recruits the tRNA into that site. One site is known as the A site, second site is known as the P site, and the third site is known as the E site. Well, the A site is called amino acyl site. In this site, it recruits a newly charged tRNA. In this site, in the A site, it recruits the newly charged tRNA into the A site. Whereas P site is the peptidyl transferase peptidyl site, where peptidyl tRNA is being formed, which is attached with many amino acids. During the process of elongation, you will see this P site is also the site for the initiator tRNA as well. Well, first tRNA which has to be incorporated for the start codon AUG will be introduced in the P site. So the P site is the first place where the initiator tRNA comes and binds. And in the next steps during the elongation, all the amino acid tRNA will entry to the A site. And from A site, it will move to the P site during peptidyl transferase reaction. E site is the place for uncharged tRNA, where the tRNA have after the process of peptidyl transferase reaction, as you know that figure. Remind that figure. I will try to get that figure now. So you will see that at the E site, the uncharged tRNA moves to the E site where no amino acid is attached, and the tRNA then goes out from the ribosome and carries out the next round of amino acylation of tRNA, where it again becomes charged with specific amino acid. So just reminding you this thing. Okay, so if you see that this reaction I have already mentioned with you, I have already talked with you about this peptidyl transferase reaction. So this is the newly amino acid tRNA. Suppose this is the A site of the ribosome. This is the A site of the ribosome where amino acyl tRNA is placed, and this is the P site. This is the P site of the ribosome where peptidyl tRNA is present, and on the side there is the E site. So what happened during peptidyl transferase reaction, that we know very well. So what happened? The ribosomes closely associate these two tRNA. Their 3' ends located very close to each other, so that the amino group of this amino acyl tRNA can cause can attack to the carboxyl group can attack to the carboxyl group, that is the carbon atom double bonded to an oxygen atom, this one of the most carbonyl terminal carboxyl terminal amino acid. So here the so this amino group is attacking to the carboxyl group of what of which amino acid? It is attacking to the carboxyl group of the most carbonyl group of the most carboxyl terminal amino acid, that is the amino acid which is associated with the tRNA in the peptidyl tRNA in the P site. The amino acid which is attached with the tRNA in the P site, there the carbonyl group is being attacked by the amino group of the amino acyl tRNA, which leads to the breakage of this bond between the tRNA, and formation of a peptide bond occurs between the amino acid found on the amino acyl tRNA with the remaining amino acids, with the remaining peptide chain of the peptidyl tRNA. And this reaction is called peptidyl transferase reaction. After this peptidyl transferase reaction, what happened? You can see very well. Now this whole tRNA have the it becomes now peptidyl tRNA, where uh, the polypeptide chain has been attached with a new amino acid to increase the length of the polypeptide. And on the other hand, the peptidyl tRNA which was before, it now become uncharged tRNA, it will be now moved to the E site of the ribosome, while this amino acid tRNA which now has become peptidyl tRNA, it will move to the P site, and creates a space in the A site, so that new amino acid tRNA can come and carry out the next round of peptidyl transferase reaction. Okay, so this is what how the whole process goes on during the process of translation. Okay, so I hope you have understood what I have said in this diagram. Okay, now let us move to the process of translation. Well, translation occurs in three main steps. So there are three steps for the translation process: initiation, elongation, termination. Just like replication, just like your transcription, translation also has been divided into three steps. So the first step is your initiation. Let us see. Initiation. Initiation of translation in prokaryotes involve attachment of small ribosomal subunit to the mRNA on the ribosome binding site. Well, ribosome binding site is, we all know that this ribosome binding site is found on the mRNA in prokaryotes, and it is complementary with the 16S rRNA of the small ribosomal subunit. That's why the small ribosomal subunit bind to the mRNA. Okay, so we are discussing about the initiation in prokaryotes. Don't forget, in eukaryotes it is little bit complicated, right? So first thing what we see that by observing the ribosome binding site, also called sign Dalgarno sequence, your small ribosomal subunit attached to the mRNA, right? Now, the small ribosomal subunit binds with the mRNA. The small ribosomal subunit binds with the mRNA in such a way so that the start codon will be in the P site where when the large ribosomal subunit join the complex. Now, the small ribosomal subunit binds with the mRNA in such a way so that your start codon, which is your AUG, it should be in the P site, because initiation during initiation process your first tRNA, which will code for methionine, which will code for N-formyl methionine actually, then it will be converted into methionine by prokaryotes. That first initiator tRNA will be placed in the P site, and for that reason the start codon AUG must be located in the P site. So that's why it is saying the small ribosomal subunit binds with the mRNA in such a way so that your start codon will be in the P site when the large ribosomal subunit joins to form that translation complex. Now this interaction, so that the the ribosome should be placed in such a way so that within the P site your start codon will be present. This interaction between the ribosomes, between the mRNA, is also catalyzed by three helper proteins called three auxiliary proteins known as the initiation factors. So the interaction for this initiation process, it is done by three helper proteins known as the initiation factors, and the name of the initiation factors involved in translation in prokaryotes are your IF1, IF2, and IF3. So these are the three initiation factors involved in translation in prokaryotes. Okay, so what is the function of these initiation factors and how these initiation factors arrange the ribosomes in such a way so that the ribosome is placed in such a way so that the start codon will be located within the P site. Okay, let us see. So these initiation factors bind to near the tRNA binding site, or it may bind to the tRNA at the tRNA binding site, either near to the tRNA binding site or within the tRNA binding site. These initiation factors will bind, that is the A site, P site, E site. These three sites are very important for tRNA to bind. Within nearby to this site or within that site, these initiation factors actually binds first. Let us see the functions of each of them. So I hope you have understood this part, what I have said. I have said that the first thing that occurs in prokaryotic translation initiation is the attachment of the small ribosomal subunit with the to the mRNA, and it is done by the interaction between 16S rRNA with the ribosome binding site. Okay, with the ribosome binding site. Okay, and after this interaction the ribosomes are placed within the mRNA in such a way so that the start codon should be located in the P site. The start codon AUG should be located in the P site of the ribosome, because initially the initiator tRNA will be binding to the P site by recognizing this start codon, and this interaction will be helped by three helper proteins are there, three auxiliary proteins are there, known as the initiation factors, IF1, IF2, and IF3, right? Now these initiation factors actually binds either near to the tRNA binding site, or directly it binds within the tRNA binding site, that is the A site, P site, and E site. Let us see now one by one. IF1. IF1 or initiation factor 1 prevent the initiator tRNA to bind to the A site. So the main purpose function of the initiation factor 1 is to prevent the binding of your initiator tRNA. Which is your initiator tRNA? Initiator tRNA is carrying which amino acid? Here, in case of prokaryotic translation, the initiator tRNA is carrying the amino acid called N-formyl methionine. It has a formyl group. Later on, that formyl group will be removed and methionine will be there. So the first amino acid which is to be incorporated to the growing polypeptide chain is your N-formyl methionine. Okay, so this IF1 or initiation factor 1 prevent that initiator tRNA which is carrying that N-formyl methionine to bind to the A site. So it does not allow the initiator tRNA to bind to the A site. IF2 then binds with IF1 and allows the binding of the initiator tRNA to bind to the P site by contacting with the initiator tRNA. IF2 with IF1 allows the initiator tRNA, which is carrying the amino acid N-formyl methionine, to bind to the P site by contacting with the initiator tRNA. IF2 has a GTPase activity. IF2 has a GTPase activity. Well, you know, GTPase activity is actually, that means it has a enzymatic activity. It is a protein that can bind with a GTP as well as it can hydrolyze a GTP molecule. So IF2 has a GTPase activity, means this protein, this IF2 protein can bind with GTP molecule. It can only be associate with the hair unless it is attached with GTP, as well as it can hydrolyze the GTP as well. So IF1 prevents the attachment of initiator tRNA to bind to the A site, while IF2 with IF1 allows the initiator tRNA to bind to the P site of the ribosome, where your AUG codon is placed on that site by contacting with the initiator tRNA. And IF2 have a GTPase activity which can hydrolyze a GTP molecule, and it is associated with GTP as well. So IF2 actually main function is to allow the initiator tRNA to bind at the P site and also prevent the other charged tRNA from associating with the small subunit. So IF2's main function is it has a GTPase activity that you will see during initiation process, and it allows the initiator tRNA, which is carrying the amino acid called N-formyl methionine, to bind to the P site. And it also, this IF2 also prevent the other charged tRNA from associating with the small ribosomal subunit, because now it is the time for initiation, and for initiation we need initiator tRNA. No other charged tRNA should be present there. So it is preventing the other charged tRNA from associating with the small ribosomal subunit, as well as it is allowing the initiator tRNA to come and bind to the P site. The last initiation factor is your IF3. IF3 prevent the assembly of the large and the small ribosomal subunits. Well, IF3 is the another initiation factor which prevent the assembly of large and small ribosomal subunits, because during initiation we have to arrange all those initiation factor one by one to the position, so that our initiator tRNA will get that position to bind at the P site, and then only your large ribosomal subunit will associate. Then before that, the IF3 has to got out from the place, because IF3 is disassembling the large and the small ribosomal subunit. So you can see here what IF3 do. It does not allow the large and small ribosomal subunit to come and bind. So it is needed for the initiation process, so that the initiation factors arrange themselves, so that our initiator tRNA can able to bind to the P site. Then only IF3 job is over, and it then goes off from the initiation complex, so that your large ribosomal subunit bind. Okay. During translation initiation, free ribosomal subunit is required. So for the initiation process of translation, a free ribosomal subunit is required. That's why IF3 binds to the small ribosomal subunit and prevent it from reassociating with the large ribosomal subunit required for the translation initiation process. So during translation initiation, free ribosomal subunit is required. IF3 is making that free ribosomal subunit available by binding with the small ribosomal subunit, so that the large ribosomal subunit will not bind, and thus they can carry out for the translation initiation process. The other factors will bind. It will place the initiated tRNA to the P site, so that it can interact with the start codon, and then thus when initiation process is complete, then only IF3 will got off from the small ribosomal subunit, so that the large ribosomal subunit will bind and then proceeds for the elongation process. Okay, so this is the function of the IF1, IF2, and IF3. What does IF1 do? IF1 prevents the initiator tRNA to bind to the A site. What does IF2 do? IF2 associate with the IF1 and allows the initiator tRNA to bind to the P site. Okay, IF2 also have a GTPase activity, which means this IF2 associate with a GTP molecule. It associate with a GTP molecule, and it can also hydrolyze the GTP molecule. And so it's also a function. IF2 also have a function that it also prevent the other charged tRNA from associating with the small ribosomal subunit, because now it's a time for initiation, not elongation. So I have to do not want to do that. It allows only the initiator tRNA to bind at the P site where the start codon is present, is being located, so that they can able to interact by the anticodon sequence found on the initiator tRNA with the codon region of the mRNA located at the P site. Okay, and IF3 function is to prevent the assemblage of large and small ribosomal subunit, because during initiation we need free ribosomal subunit, and IF3 binds to the small ribosomal subunit, so that uh, the other initiation factors can allow the process of initiation, and when the whole process is complete, then only IF3 will goes off from the small ribosomal subunit and allows the large ribosomal subunit to bind and carries out the elongation process of translation. Well, the initiator tRNA that I was talking about in case of prokaryotes is charged with N-formyl methionine. So the amino acid which attach with the initiator tRNA is called N-formyl methionine, and we call them in this way: F-met tRNAi F-met. F-met means formyl methionine dash tRNAi, I for initiator tRNA. In the above we write F-met, that means formal methionine. So this is the in this way we name the initiator tRNA. Now in eukaryotes, we don't have N-formal methionine as our first amino acid. The initiator tRNA is charged with methionine only, and that's why the name of the initiator tRNA in eukaryotes will be called as met dash tRNA I for initiator tRNA, met for methionine. Okay. So for the prokaryotes, the initiator tRNA is N-formal methionine, while for the eukaryotes, the initiator tRNA is your carrying the amino acid methionine. Binding of the formal methionine to the small ribosomal subunit will be facilitated by the interactions with the IF2 bound with GTP and base pairing between the anticodon and start codon of the mRNA. So, how does the initiator tRNA, which is carrying the amino acid formal methionine, how the initiator tRNA binds into the ribosome in prokaryotic initiation process? It's by two ways. The first way is anticodon and start codon of the mRNA. The start codon is placed on the P site of the ribosome, as I have said, and the anticodon sequence of the initiator tRNA is placed on the P site also, so it interacts with the start codon and binds to the P site. Secondly, IF2 IF2 bounded with GTP helps to bind that initiator tRNA to bind to the P site. So because of these two reasons, initiator tRNA very slowly bind to the P site of the ribosome, because at the P site there is your start codon, and by anticodon codon pairing they bind. And on the other hand, your IF2 with their GTP activity, it helps that initiator tRNA to bind to the P site of your ribosome. Now, when the start codon, that is AUG, and the initiator tRNA base pair with each other, the small ribosomal subunit undergoes a change in their structure. There will be a conformational change in the structure of the small ribosomal subunit when the start codon and the initiator tRNA base pair with each other, and this altered conformation result in release of the IF3. So simple thing, when your initiator tRNA properly binds to the P site of the ribosome, there will be a change in the conformation, there will be change in the structure of the small ribosomal subunit, which causes the release of IF3. And we know very well IF3 was causing the disassembly of the small and the large ribosomal subunit. So in the absence of IF3, the large ribosomal subunit is free to bind to the small subunit, okay, along with IF1, IF2, mRNA, and your initiator tRNA. So in the absence of IF3, the large ribosomal subunit binds with the small ribosomal subunit, and now the small ribosomal subunit is having that IF1, IF2 with GTP activity, attached with GTP, mRNA with initiator tRNA. Now, when large ribosomal subunit binds, when your large ribosomal subunit binds, it will stimulate the activity, that is your GTPase activity of IF2 GTP, causing it to hydrolyze GTP. That means to say, what we have said first, I have said that when the initiator tRNA bind to properly to the P site, some conformational change occur in the structure of the small ribosomal subunit which causes to release IF3. Okay, when IF3 got released, the large ribosomal subunit binds. When the large ribosomal subunit binds, the GTPase activity of IF2 activates. That means IF2 now cause hydrolysis of its own bound GTP. IF2 has been attached with a GTP molecule, but when the large ribosomal subunit binds, IF2 catalyzes its GTPase activity. It hydrolyzes this GTP molecule, and the resulting IF2 is now having GDP attached. Well, IF2 can only attach with the ribosome when a GTP molecule is present, but now as IF2 has hydrolyzed the GTP, IF2 can has a less affinity for the ribosome and initiate a tRNA. That means simply IF2 GDP has a less binding affinity with the ribosome, as well as it has less binding affinity with the initiator tRNA. Thus, it causes to release that IF2 GDP as well as IF1 from the ribosome. So what's happened? A great thing has happened when the large ribosomal subunit bind. The GTPase activity of IF2 have occurred. IF2 then what it does, it hydrolyzes its own bounded GTP and makes GDP. So when IF2 hydrolyzes the GTP to form GDP, IF2 cannot able to interact properly with the ribosome, as well as it cannot able to interact with the IF1. Why? Because it has less affinity. IF2 with GDP has a less binding affinity with the ribosome. It's simply to say that IF2 can only attach strongly with the ribosome with the IF1 when it is bound with GTP. But as IF2 has hydrolyzed the GTP as a result due to the binding of large ribosomal subunit, so now IF2 can no longer attach with the ribosome with the IF1, so it get released from the initiation complex. So thus the ribosome mRNA complex is now ready to accept a charged tRNA into the A site, because after IF2, IF1 also dissociate from the ribosome. So almostly all, not almostly, all the initiation factors have been removed now. Now your ribosome mRNA complex is ready for the elongation process so that they can accept a charged tRNA, that means a tRNA having an amino acid bound. So they can use this charged tRNA to bound to the A site of the ribosome and start our polypeptide synthesis. So very wellly it has been said about the initiation. So if you see through diagrammatic view, we will able to understand. So this is that interaction that I have said. This is that interaction that I have said. This is the small ribosomal subunit. It attach with the large ribosomal subunit. It attach with the mRNA, sorry, and it is attaching with the mRNA in such a way, the small ribosomal subunit is attached with the mRNA in such a way so that the start codon should be located within the P site. Okay, this interaction is shown by the 16S rRNA with the ribosome binding site. Okay, so don't need to worry. Okay, now this is a more clarified diagram which is shown that how the initiation factors associate with the small ribosomal subunit? Well, you can see IF3 has been attached. IF3, what it does, you already know. IF3 normally, what does it do? IF3 uh, prevents the assembly of the large and the small ribosomal subunit, because a free ribosomal subunit is required for your translation process to begin. That's why IF3 bind at the E site. IF1 and IF2, they are allowing the initiator tRNA to bind to the P site, not to the A site, because initiator tRNA must bind to the P site where your start codon is present, AUG is present, right? Okay, so this is the model of initiation factor bind to the 30S ribosomal subunit, which has been shown. You can also study these parts, which I have shown. I have already discussed with you. You can see what it has been said. IF1 prevents the initiator tRNA from binding to the position portion of the small subunit that will become a part of the A site. That means IF1 prevents the initiator tRNA to bind to the A site, directly speaking. It allows the tRNA to initiator tRNA to bind to the P site. IF2 has a GTPase activity. That means it can hydrolyze GTP. It can also bind with the GTP molecule. So it has a GTPase activity. IF2 interacts with the three key components of the initiation machinery. IF2 interacts with the small ribosomal subunit. IF2 interacts with the IF1, and also IF2 interacts with the initiator tRNA, which is carrying N-formyl methionine. So by interacting with these components, IF2 facilitates the subsequent association of this initiator tRNA with the small ribosomal subunit in the P site. And IF2 also prevent the other charged tRNA from associating with the small ribosomal subunit. This thing I have already discussed with you. The last is your IF3. IF3 binds to the small subunit and prevent it from reassociating with the large ribosomal subunit or from binding charged tRNA as well. Because for initiation process we require free small subunit, we require free ribosomal subunit to carry out our initiation process. So binding of IF3 is very critical for a new cycle of translation. IF3 become associated with the small subunit at the end of the previous round of translation, when it helps when it helps to dissociate the 70S ribosome into large and small subunit. Well, you will see this function of IF3 again at the termination part. During the termination of your translation process, you will see that IF3 again comes and disassemble the large and the small ribosomal subunit. Okay, after identification of the after the release factors, after the release factors of the termination pathway comes. Okay, of the termination machinery comes, so that they can carry out the next round of translation. So this is what I have said. This is that picture how the initiation factors associate during the initiation of translation. Okay, so this image is the whole image is look like this, and this is the image that what we have studied throughout the initiation process. You can see what it happens. Let us see. So we can see through picture by picture. You can see here, you can see here, this is the your this is your 70S ribosome, the whole ribosome, the prokaryotic ribosome that you can see. It disassemble into large and small subunit by the help of IF3. So IF3 bound to the E site of the small ribosomal subunit, and it allows the disassembly of the ribosomes. So the large subunit separates from the small subunit. Now what happened, you can see that the other initiation factors like IF2, IF1 with GTP, because IF2 has a GTPase activity, it can only attach with the ribosome unless it is attached with GTP. So you can see IF1, IF2 with GTP and IF3 have positioned their place so that the P site, so that within the P site it allows the charged tRNA to come and bind, to come and bind. Well, you can see the initiated tRNA, which is your F-met tRNA IF-met. This initiated tRNA is allowed now to bind to the P site of the small ribosomal subunit. So now it is called as 30S initiation complex. This is known as now the 30S initiation complex. Now what happened? You can see the this ribosome is now associating with, already it is showing that it already associated with the mRNA, but in this diagram it shows that it is associating with the mRNA. So during association of the so the large ribosomal subunit now tries to associate, and for this that the initiation factor 3 has to comes out. Okay, actually how large ribosomal subunit binds is because the small ribosomal subunit, when the initiated tRNA properly bind to the P site and properly interacts with the start codon of the mRNA at the P site, then there will be a conformational change in the structure of the small ribosomal subunit, which allows the release of IF3, and that causes the binding of large ribosomal subunit, because we know that IF3 was disassociating the large and small ribosomal subunit. Now, due to now when the large ribosomal subunit binds, then another event occurs. This the GTPase activity of the IF2 activates, and it hydrolyzes its own bound GTP and becomes IF2 GDP. IF2 GDP, IF2 GDP. Sorry. Okay. IF2 GDP has less affinity to bind with the ribosome. Also, it has less affinity to bind with the tRNA, initiated tRNA, and also with the IF1. So, IF2 also goes out from the initiation complex, along with IF1 also goes out. Thus, your 70S initiation complex is now ready for the elongation process to proceed. You can also see that the A site is now, there is a space in the A site, so that your new amino acid tRNA can come and bind to that A site by proper codon anticodon pairing to carry out the elongation process. Okay. So this was about the translational initiation in prokaryotes. One more thing that I have not mentioned is about the N-formal methionine. Let us see. As N-formal methionine is the first amino acid to be incorporated into the polypeptide chain, we might think that all prokaryotes protein have a formal group at their amino terminus, right? But this does not happen. This N-formal group will be removed later on by another enzyme called deformylase. Deformylase removes that formal group from the amino terminus of the polypeptide chain during the process of translation or after the synthesis of the polypeptide chain. So this has to be remembered that the N-formal methionine, although we know that this is the first amino acid which is incorporated to the growing polypeptide chain in prokaryotes, but still if you observe the protein sequence in prokaryotes, if you see the prokaryotic proteins, their N-terminal group does not have any formal group. So where does the formal group go? the formyl group is being removed by an enzyme called deformylase. During the process of translation or after the polypeptide chain synthesis, deformylase works. Okay, so this is the structure of the N for methionine and N formyl methionine. The difference is that this formyl group is present. Okay, this formyl group will be later removed by deformylase enzyme. Okay, the same figure has been given here. Now we will discuss about the initiation of translation in eukaryotes, right? Well, initiation in eukaryotes is bit complicated. Really, it is complicated, but we have to listen it carefully, step by steps, because there are so many initiation factors involved in initiation in eukaryotic translation process. But we will discuss some of the main factors here, not all of them. Okay, because some are related with the research now, some are not been their function has not been characterized. Let us see some of the important initiation factors involved in translation initiation in eukaryotes. So, initiation of translation in eukaryotes. Eukaryotes also require many more auxiliary proteins, which are the initiation factors. Auxiliary mainly means helper proteins to initiate the translation process. The ribosomes are dissociated into large and small ribosomal subunit by the action of initiation factor called EIF3 and EIF1A, which is similar to IF3 and IF1 in case of prokaryotes. So what does it mean? It's pretty straightforward. It is saying here eukaryotes need many more initiation factors. Now, to dissociate the ribosome into large and small subunit, we need initiation factor EIF3 and EIF1A. In case of eukaryotic translation initiation, we need EIF3 and EIF1A to disassociate the ribosomes into large and small subunit. This EIF3 and EIF1A is analogous, or you can say similar to the IF3 and IF1 found in prokaryotes. So I have written again for you easy to remember. EIF3 and EIF1A causes disassembly of the ribosome into large and small subunits. Next, there is two GTP binding proteins in eukaryotic translation initiation. Well, we have seen in prokaryotic translation initiation there was only one initiation factor known as IF2, which has a GTPase activity. But here in case of eukaryotic translation initiation, there are two GTP binding proteins. So next two GTP binding proteins known as EIF2. I have written here GTP, that means it is attached with GTP, and EIF5B. It is also a GTP binding protein. Both of them have a GTPase activity that mediates the recruitment of the initiator tRNA. So the initiator tRNA is recruited, is allowed to bind to the P site by the help of these two GTPase activity proteins known as EIF2 and EIF5B. EIF5B is analogous with the IF2. So the structure of EIF5B is similar with the IF2. Okay, it is similar with the IF2. Okay, now this EIF5B with GTP associate with the EIF1A and helps to recruit a complex of EIF2 GTP and initiator tRNA to the small subunit. So here two GTP binding proteins. Here two GTP binding proteins come into play. EIF5B and EIF2. EIF5B, it associate with the EIF1A and allows the initiator tRNA to bind to the ribosome, whereas EIF2 already associate with the initiator tRNA, which allows them to bind to the ribosome. So together EIF5B and EIF2 position the initiator tRNA in the P site, and this leads to the formation of 43S pre-initiation complex. So very well here more initiation factors are involved. Well, we have seen that the initiation factor called EIF3 and EIF1A disassociate the ribosome into large and small ribosomal subunit. Secondly, we see here two GTP binding proteins here, EIF5B and EIF2. EIF5B actually associate with the EIF1A and helps to recruit the initiator tRNA to bind to the P site. Well, EIF2 already associate with the initiator tRNA and allow and facilitates that binding to the P site. So together EIF5B and EIF2 position the initiator tRNA that carries the amino acid methionine in the P site, and this leads to the formation of 43S pre-initiation complex. So the 43S pre-initiation complex consists of which initiation factors? 43S pre-initiation complex consists of EIF3, EIF1A, which cause disassembly of the ribosomes into large and small ribosomal subunit. It also consists of EIF5B and EIF2. Both of EIF5B and EIF2 is a GTP. They are GTP binding proteins. That means they can only attach with the ribosomes and to the other initiation factors when it is bound with GTP. It has a GTPase activity as well. That means it can hydrolyze the GTP as well. And these EIF5B, EIF2, EIF3, EIF1A, they positioned the initiator tRNA and the small they position the initiator tRNA to the P site and the small ribosomal subunit where all this assembly occurs together form the 43S pre-initiation complex. Okay, now this 43S pre-initiation complex that I was talking about, that is, it is composed of EIF3, EIF1A, then the two GTP binding proteins EIF5B and EIF2 um, together form that 43S pre-initiation complex. This 43S pre-initiation complex that is observed on the small ribosomal subunit has to be recognized, and is recognized how it recognizes that mRNA. Well, I have said before that normally ribosomes recognize the mRNA by observing the 5 prime cap. So by observing the 5 prime cap found at the 5 prime end of the eukaryotic mRNA, the ribosomes bind. Okay, and actually not only the ribosomes fast together with the initiation factor and the initiator tRNA makes that 43S pre-initiation complex. then it binds to the uh, mRNA. Okay, by in through the by observing the 5 prime cap sequence. Okay, in Bengali I have written, just go through this. Okay, so now we can see more other initiation factors are also involved for the whole initiation process. Well, we can see on this side the 43S pre-initiation complex first formed, and after the formation of 43S pre-initiation complex within the ribosome, this whole complex begins to recognize the 5 prime cap, and then it binds to the eukaryotic mRNA. On the other hand, there is also some more initiation factor that recognize the 5 cap 5 prime cap region as well. Well, EIF4F, this is another initiation factor found in eukaryotic translation process which recognize the 5 prime cap of the mRNA. EIF4F is made up of three subunits. EIF4F, this initiation factor is made up of three subunits: EIF4E, EIF4G, and EIF4A. So EIF4F, it is another initiation factor that recognize the 5 prime cap of the mRNA actually, and this initiation factor has three subunits: EIF4E, EIF4G, and EIF4A. And out of these three subunits bind directly to the 5 prime cap of the mRNA, while the one of these three subunits bind to the 5 prime cap of mRNA, while the other two subunits they bind non-specifically to the associated mRNA. So EIF4F, they have three subunits, and these subunits associate with the 5 prime cap, and one of the subunits of EIF4F, it is showing that E, that means EIF4E directly binds with the 5 prime cap, while the other two subunits of EIF4F, which is your EIF4G and EIF4A, they loosely associate with the 5 prime cap. They little bit loosely associate with the or non-specifically bind to the 5 prime cap. Okay, and due to this association of EIF4F, this complex allows binding of EIF4B. Well, one by one, more initiation factors come and play here. So I have said before, long before, about the 43S pre-initiation complex, which has been formed by the EIF1A, EIF3, EIF5B, EIF2, which allows the initiator tRNA to bind to the ribosome. Okay, that's over. On this hand, to identify the mRNA, your ribosome has to bind to the mRNA. It has to recognize the 5 prime cap. Okay, so but before that, long before that, other initiation factors interacts to this 5 prime cap, like I have said about EIF4F. EIF4F recognize the 5 prime cap of the mRNA. It has three subunits that I have mentioned, and one of these subunits, that is your EIF4E, they directly bind to the 5 prime cap. You can see here I have little bit short in draw. I've drawn a diagram of EIF5E, just a round figure where it associate with the 5 prime cap, right? It associate with the 5 prime cap. while the other two subunits they non-specifically bind with the 5 prime cap, the EIF5G, EIF4G, and EIF4A. They non-specifically bind to this cap portion. Okay, this allows binding of another initiation factor called EIF4B. EIF4B activates an RNA helicase activity in one of the EIF4F subunits. So within the EIF4F subunits, there has a RNase helicase activity. What does EIF4B do? When EIF4B is recruited, it binds and activates an RNA helicase activity of the EIF4F subunits. Normally, we all know RNA, here mRNA is single strand. But what does this RNA helicase will do? Helicase normally unwind the double stranded DNA we have seen. But what about RNA helicase? RNA is normally single strand. How RNA helicase will work here? Well, there are some regions of the mRNA which fold onto itself to form secondary structures. Just like hairpin loop I have said before, due to base complementary regions, they often fold onto itself and form secondary structures. We have to remove these secondary structures by breaking those hydrogen bonds. We have to unwind them, and that's why RNA helicase activity comes. EIF4F actually have they activate that RNA helicase activity, which is found on one of the EIF4F subunits. And this RNA helicase will unwind those secondary structure of the mRNA, like the hairpin that has been formed due to folding onto itself. The mRNA has folded onto itself due to inverted repeat sequence that form this type of structure, secondary structure. It has to be removed out. Otherwise, it will create a problem during translation process. So that's why this activity is needed, and it is recruited, actually it is activated by EIF4B, and this RNA helicase activity is located in one of the EIF4F subunits. Okay, it has not been mentioned which subunit is responsible for the RNA helicase activity. This RNA helicase activity of one of the EIF4F subunits will then uh, unwind the secondary structure if present on the mRNA, like hairpins that may be formed at the end of the mRNA, and allows the binding of the prepared 43S pre-initiation complex to the mRNA. So now, as the hairpin, as the secondary structure of the mRNA has been removed, plain mRNA is there now. Now it is the time for recruitment of that 43S pre-initiation complex to the mRNA. It will recognize the mRNA because already the other initiation factors like EIF4F have interacted with the 5 prime cap. So by interaction, it will bind. Okay. Now once bound to the mRNA, this 43S pre-initiation complex now moves in a 5 prime to 3 prime direction, and which is called scanning, and it will move until it recognizes the start codon. So once your 43S pre-initiation complex has attached with the mRNA, it will now move from 5 prime to 3 prime direction, which is purely a ATP dependent process until it recognizes the start codon AUG. And when it recognizes the start codon, obviously there will be correct base pairing between the codon and anticodon sequence, and due to correct base pairing between the tRNA anticodon sequence with the codon regions of the mRNA, some conformational changes occur. It will that causes release of the initiation factors like EIF3, and EIF3, as it become lost, it allows the binding of the large ribosomal subunit. As usually, when the large ribosomal subunit binds, so again I'm repeating, loss of EIF3 allows the binding of large ribosomal subunit, and when large ribosomal subunit binds, it stimulates the GTPase activity of EIF5B and EIF2. Just like in case of initiation of translation in prokaryotes, when the large ribosomal subunit binds, IF2 activates its GTPase activity, it hydrolyzes its own bound GTP, becomes GDP, and it becomes disassociate from the initiation complex. Here also, when the large ribosomal subunit binds, it causes the GTPase activity, it stimulates the GTPase activity, it activates the GTPase activity of EIF5B and EIF2. It hydrolyzes its own bound GTP, and thus without GTP molecule it cannot able to bind with the initiation complex. It results in shedding off, and other initiation factors also get shed off from this from this from the ribosome. Okay, so I think you have understood what I have said. So after the formation of 43s pre-initiation complex, we all know that AIF4F recognize the 5 prime cap of the mRNA. Okay, and one of the subunits of EIF4F uh, directly binds to the cap, while the other subunits called EIF4G and EIF4A, they they non-specifically binds to the cap. This interaction of EIF4F allows the binding of EIF4B. EIF4B activates the RNA helicase activity in one of the EIF4F subunits. We don't know which of the subunits of EIF4F have that RNA helicase activity, but the main function is that this RNA helicase will unwind all the secondary structures of the mRNA if present, like the hairpins which may be formed due to base pairing of itself. If there is inverted repeat sequence, it causes them to form that type of secondary structure. Okay, and then after that it will allow that 43 pre-initiation complex to bind to the mRNA. Now when 43s initiation complex bind to the mRNA, this 43s pre-initiation complex will now move from 5 prime to 3 prime direction in search of a start codon. And when it recognizes the start codon, proper codon anticodon base pairing occurs between the initiator tRNA, which was already present in that 43s pre-initiation complex, and it causes some structural change which leads to release of EIF3. Well, EIF3 and EIF1A both are causing disassembly of the ribosomal subunits, we have said before. But due to removal of EIF3, it allows binding of the large ribosomal subunit. So when large ribosomal subunit binds, it stimulates the GTPase activity of the EIF5B and EIF2. It stimulates the GTPase activity of EIF5B and EIF2, which result in shedding of other initiation factors. So when large ribosomal subunit binds, when the large ribosomal subunit binds, it cause it stimulate the GTPase activity of EIF5B and EIF2, and we all know that these GTP binding proteins can only associate with the ribosome, can only associate with the other initiation factors when it is attached with GTP. But due to hydrolysis of the GTP molecule, what happened? It no longer associate with the ribosome because of weak interaction, and it shed off. Okay. Now, a strange thing I want to say that that EIF4G that I have said, one of the subunits of EIF4F, right? I have said now that EIF4G is also a subunit of EIF4F. Well, you can see here it has three subunits. So this EIF4G I have given star mark. Why? Because EIF4G has a special property. And what is that special property is that EIF4G can interact with the polyatal. Not polyatal, it can interact with the poly A binding protein. Poly A binding protein normally observed on the poly A tail. It coats that poly A tail actually. So this poly A binding protein, which I have drawn, I have tried to draw this figure. This poly A tail that you can see here, you are seeing here, it is associated with poly A binding protein, which is shown in red here. Okay. Now, EIF4G, which is one of the subunit of EIF4F, it can interact with this poly A tail, and that's why circular mRNA is being observed here, right? So, EIF4G can interact with the poly A binding protein. That this poly A binding protein coats the poly A tail, and due to interaction of EIF4G with the poly A binding protein, it results in circular mRNA configuration. Okay, so a protein bridge has been created between the EIF4G and the poly A binding protein located at the 3' end, which leads to a circular configuration in the mRNA. Okay, in this way, the EIFs are not IF, EIF. IF is related with protein initiation factor, but when I say EIF, E for eukaryotic, I for initiation, F for factor. So these EIF are closely associated with the 3' end of the mRNA with its poly A tail and contributes to efficient translation of the mRNA. So in this way, your interaction between the EIF with the poly A tail with the 5' cap is made to make a circular configuration of the mRNA figure. Okay. So this is the what we have all discussed about for the eukaryotic initiation of transcription process. Okay. So here you can see the in two different figures has been shown here. So you can see here how the 43S pre-initiation complex occur. Let us see first in this spot for 43S pre-initiation complex. You can see the large and small ribosomal subunit. It is getting disassociated because of these two initiation factors called EIF1A and EIF3. And then what happened? Then your EIF5B, if I zoom it, you can able to see EIF5B associate with the EIF1A, and EIF5B with EIF1A is recruiting the initiator tRNA to bind to the P site of the ribosome. Okay, and you can see another GTP binding protein called EIF2. EIF2 is also a GTP binding protein just like EIF5B, but here EIF2 associate with the initiator tRNA and is recruiting that initiator tRNA to be placed on the P site of the ribosome. So two GTP binding proteins found on the eukaryotic initiation factors of translation. We see EIF1A also, EIF3 also, which is causing disassembly of the ribosome into large and small ribosomal subunit. So this is your 43S pre-initiation complex what is about. Now let us see the other thing. You can see here that within well, eukaryotic mRNA do not have a ribosome binding site, but they have a 5' cap. This 5' cap is recognized by a translational factor, sorry to say, translational factor known as EIF4F. So EIF4F EIF4F has three subunits called EIF4E, which you can see here. It directly interacts with the 5 prime cap. EIF EIF4G, you can see here EIF4G. It do not directly interacts with the 5 prime cap. In non-specifically, it binds to the 5 prime cap, and you can see also EIF4A. You can also see EIF4A here, right here. Okay. Now, this recruitment of EIF4F allows the binding of EIF4B, and when EIF4B binds, it activates an RNA helicase activity within the EIF4F. So there is one of the subunits of EIF4F which has a RNA helicase activity, and that activates by EIF4B. So due to the RNA helicase activity, as you can see here, a hairpin loop has been shown, and this occurs due to complementary base pairing between themselves. It may occur within the mRNA, within the tRNA. Well, we can see in the tRNA, so much of base pairing occurs, folded and loops like structure occurs for their stability. Here also, mRNA tries to become stable so that they become, some areas they have folded configurations as well. This RNA helicase activity is meant to remove these hairpin configurations, remove these secondary configurations, so that linear mRNA can be prepared. And now your 43 pre-initiation complex will come and bind, come and be recruited to the mRNA. Now, when it is recruited to the mRNA, then what happens? This 43 years pre-initiation complex will move. It will move, it will scan. We can say scanning also. It will move in the direction towards the 3' in search of a start codon. When it identifies the start codon, when it identifies the start codon, proper codon anticodon base pairing occurs between the initiated tRNA and the start codon found in the mRNA, which causes the initiation factor 3 to goes off from the pre-initiation complex and recruitment of the large ribosomal subunit. Now, when the large ribosomal subunit binds, it stimulates the GTPase activity of EIF5B. And when EIF5B have stimulated the GTPase activity, that means it hydrolyzes its own GTP. It hydrolyzes its own bounded GTP. That, and you all know that this GTP binding protein can only associate with the ribosome here, can only associate with the other initiation factors, even with associate with the initiated tRNA, unless it is attached with GTP. When it is not attached with GTP, it has a weak affinity for binding, and that's why they will be removed off from the 43S pre-initiation complex. So we can see EIF3, EIF4B, EIF2, even the EIF5B will be removed off from this pre-initiation complex and allows that mRNA, allows this ribosome to be proceed for the translation elongation process. Okay, this is the structure that I have drawn, that well, a circular configuration of mRNA can be observed. A circular configuration of the mRNA can be observed. We can see here that EIF4F, this is your EIF4F. It's one of the subunits of EIF4F. You can see EIF4G. It can interact with the poly A binding protein. Well, poly A binding protein found on the poly A tail, which coats the poly A tail. So by this interaction between the EIF4G with the poly A binding protein, it causes a circular configuration of the mRNA, right? So it has been written here. Let us see. It has written here that circularization Circularization is proposed to be mediated by interaction between EIF4G subunit of EIF4F and poly A binding protein. The EIF4G subunit of EIF4F and your poly A binding protein is causing that circularization of DNA of your prokaryotic mRNA of eukaryotic mRNA. Sorry. Okay, in the next lecture, I will discuss about the elongation process. So, just after this lecture, I will go for elongation now. So, I am to end this process because it's over more than 1 hour, and this video will be permitted to 2 hours only. So, I have to do a next video related with the elongation. Okay.